Good. Then I think I'll start with welcoming everyone to today's MtL Lecture Series, a lecture series where we introduce the fellows of the Max Planck School MtL, which are actually distributed all over Germany. So we have currently 14 locations where our fellows are based. And today is my great pleasure to welcome Laura de Laporte, which is in Aachen at the DWI. And yeah, we have two questions prepared as always in order to get to know you a little better before your presentations. And this is what I would start with. And my first question to you would be, can you tell us about your current role at the Max Planck School MtL and how your work contributes to the broader MtL field?
Yeah, so I guess my role is that we do both teaching and research. So I'm one of the professors who is teaching also an introduction to chemistry course for the students. And I think the students have very broad interdisciplinary backgrounds. So I think it's very good to do this to get also everyone like on a certain basic level of the different disciplines, because that's also matter to life. I think it's very interdisciplinary, combining chemistry, physics, biology, engineering. So I think it's good to do that. And then besides that, of course, we do a lot of research, and in our research, which in a way then contributes also to matter to life, is we rebuild like tissues and cellular constructs in vitro, but also to repair tissues in vivo. To basically create life again, right, or like outside of the body, and then study a lot of biological questions. And in that sense, we contribute to the program because we host a lot of students at the different levels. There's undergraduate students that sometimes come visit us. We then have rotation students who want to kind of learn about what we do, who are here then for like about 3 months together usually with another lab. And then we have the master's students who can come for like 6 months and then really do like a master's thesis. And then finally, if people really, really like our topic, they can also stay potentially as a PhD student and to then really do like a 3-year PhD in our lab. And so far we've hosted a lot of students here at the DWI, and we're very happy to do that.
Mm-hmm. And I think the students also have been very happy to visit you in Aachen. At least that's the feedback I got. And with this, I come to my second question. What are some of the most significant challenges you've encountered or you see in your current career so far, and how did you address them?
Yeah, so first of all, I talk with a lot of young researchers who are pursuing an academic career, and I realize how tough it is, right? And like that there's just very limited positions. So I would also like to tell everyone, like, yeah, don't give up, but also think about other plans necessarily, because there are different career options as well. Because I think besides needing very unique ideas that really set you apart from other people, which is hard, right? It's not easy to always come up with ideas that no one else has thought about. Yeah, besides that, I think you also need some luck and some people who really support you. And in that sense, I think I've been lucky to be able to do this and to be now in the position that I am. But I can tell you, I also had a plan B and a plan C, and so but now so there were a lot of challenges along the way, I would say, but now and I was talking to Jan before, one of the main challenges for me personally is that sometimes we are really interested in also translating certain things into the clinic or into industry, and I think this is very challenging because we as scientists are really not equipped, or at least I'm not equipped to do that, like all these regulatory aspects, and also like the large amounts of funding that is needed to translate something, especially also into the clinic. So this is something that I'm now looking into more and learning more about now, and I think it's a huge challenge, a huge mountain we have to go across. But just in that sense, I would like to also mention that steps are being taken. For example, here in Aachen, we now opened a new building between the DWI and the Uniclinic with clean rooms in the basement to do manufacturing according to good manufacturing practice, so GMP. and also establishing a team to help scientists to be able to do translation when a product would be ready to do that, and potentially then also test in the clinic in small clinical trials like these initiator or investigator-initiated trials is what they're called. And yeah, so this is something that would be my big dream, being in this position to eventually actually make a difference into the clinic. And that's what now is my biggest challenge.
Thank you very, very much. All the best for this challenges. And now we're getting really excited, I think, to hear about your research and what you're actually working on. And this maybe can translate then in something in the clinics later on. So I'm handing over to you. Everybody who has questions, please use the question and answer bubble on the bottom of Zoom. And I will read them out to Laura at the end of the talk, and she will gladly answer all the questions you might have.
I'm going to look at now. Wait, which screen?
Start sharing. Now we see PowerPoint.
Okay.
Yes. Yes.
So you see the slides, right? And the normal good
Yes,
mode?
it looks perfect. Yeah. So
Okay.
here's the distribution of the MtL Life School.
Yes.
Good. Yeah. With this, I would say stage is yours, and we're looking forward to hear about synthetic molecular and colloidal building blocks to overcome challenges in tissue engineering.
Yeah, so Jan, thank you very much, and thanks for everyone in the virtual room. So for those who are international and maybe don't know Germany so well, I am currently here in Aachen. So we are very close to the border with Belgium and the Netherlands. We even have a point where you like seeing the 3 different countries. So it's a very nice location to be in, and so you're if you're interested, you can always contact us to potentially also work here for a little bit. So yeah, I added a word to my title, which is injectable, and I will immediately dive into this, why I'm interested, and the whole group is working on injectable materials. And so the first original reason why we started making injectable materials was to do in vivo regeneration in places where you don't really have space. So places where you need something that you can inject in a low invasive manner and that would form a 3D construct in the body after you inject it. So to not create any more damage. For example, spinal cord, cartilage, heart, there's many examples where you don't really can cut a piece out or something to inject something. And as we were developing these materials, and also as I already mentioned, it's very challenging to finally go into the clinic, we realized that also we could use a lot of these building blocks that are injectable to pipette and to also use automated robotic pipetting systems, and to then really in high throughput combine them with cells and make 3D mini tissues. And in our case, we would like to really make human mini tissues in order to have an alternative for drug testing and other toxicity testing besides using animals. And not just to reduce animals, but also because we believe that human mechanisms can be very different compared to animals. And so in this case, you would have also human tissues. And so this is now more like a new field we're going into, and this is then all like in vitro work and not in vivo. And finally, if you want to create more complex tissues in vitro, instead of these like high throughput systems and these multi-well systems, we also started working in the field of bioprinting, because we think that a lot of the materials we have been developing could potentially also be useful here. So we don't only use macromolecules that we then could inject, and they would cross-link into some kind of hydrogel, which can very well mimic also the extracellular matrix. It can take up a lot of water. They're relatively soft, and you can modify them mechanically, but also biochemically with different peptides or different bioactive molecules. We also work a lot with microgels, and specifically microgel rods. And so what is so special about this? In a way, the biggest difference between a macromolecule and one of these microgel rods is that they're 1,000 to 10,000 times larger. And because they're larger, you can create very different structures. So they're still small enough to be injected, but now if you would interlink them, you can create much larger pores compared to a normal hydrogel, which usually has pores in the nanometer range. If you now interlink these microgel rods, you can have pores from like tens to hundreds of micrometers, so that cells can easily grow in, have a lot of cell-cell interaction, and then form these tissues into these constructs. And so the second problem in tissue engineering is that a lot of our tissues are actually structured, and we believe that the structure is very important for their function. But a lot of these hydrogels, if you inject a liquid with these macromolecules and they cross-link into a construct, there's no orientation or guidance into these constructs. And so therefore, we started adding also these microgel rods, which are magnetic, to then align and create structure into these hydrogels that are still injectable. And then the third part that I will talk about today is the fact that we move, right? Our heart is beating, we have a lot of movement in the body. And so we also wanted, especially also in our in vitro models, we wanted to study how movement can have an effect on cells, but also certain pathologies, or also have an effect on stem cell differentiation into specific human mature cells. And so these are the three topics that I would like to cover today. And we will start with the interlinking of the microgels to make these larger microporous constructs. And so this was a bit inspired also by the work of Tatiana Segura. She was making spherical microgels and then interlinking spheres to also create larger pores. But there you're a little bit limited in how big you can make the pores, because the only way to make a bigger pore is to make a bigger sphere, but then you have more synthetic materials. So we thought, what about interlinking rods? Because then by also changing the aspect ratio, we can now have bigger pores. And so you see, in this case, we have red and blue microgels. They have different chemical functionalities. I will explain later. And then they interlink chemically together and they form these large, stable constructs now having these larger pores. So just to first show you how we make them, we use a lot of microfluidics. And here you see droplet microfluidics. And basically what you do is you have a polymer solution, in this case, a free radical, for free radical polymerization with an initiator. And then you have a continuous phase, which cuts the dispersed phase into droplets. And then you can shine UV light onto the droplet in order to cross-link the polymer. So this has to happen very fast because the droplet is only in this UV spot for a very short amount of time. And you want to basically freeze the shape, because if the cross-linking is not enough and the droplet comes out of the microfluidics, it will snap back to a sphere. So you need very fast chemistries, and then you see that depending on the size of the droplet, you can make a shorter droplet or a longer droplet, and you can get these different aspect ratios. And you can also see you can make them very soft, so depending on the polymer concentration. So yeah, we can tune not only the stiffness, but also the diffusion of these microgels by changing the polymer you initially use. You can use like linear reactive polymers or star molecules, and really by tuning also the ratio of these different precursor polymers, you can change the stiffness. This is measured via nano indentation. But you can also change the stiffness independently from the diffusion into the system. And so this is sometimes very interesting depending on which biomedical application that you are working on. If you're also using them to deliver drugs or to have like certain cells interacting with these microgels, you can tune this by using different polymers. So if you now want to interlink these microgels, you need reactive groups. And this we realized by adding different monomers to the polymers. And in this case, we use or an aminoethyl methacrylate, which then gives you an amine, or we added glycidyl methacrylate to have this epoxy group. And so we would have the red and the blue microgels with the different functionalities, and then we would mix them. And then we could build these constructs. And if the light is off, you see that these rod elongated droplets will just snap back to a sphere. And so, yeah, we first looked how much of this co-monomer do we need to actually interlink the microgels. And we see that we actually needed a very high concentration, for example, of this amine. And this was because we noticed that if we don't add enough of it, that we had a lot of the functionalization into the network, but not on the surface of the microgel. And only when we had the highest concentration, we had the functional groups also on the surface of the microgel. And of course, if you want them to interlink, they need to be on the surface. And this is kind of easy to understand that you need this, but chemically it's not always that easy. So we sometimes really struggle if we modify microgels to really also get it on the outside. What you can do also is think about post modification of the network, but then you need enough functional groups to do post functionalization. So yeah, it's sometimes a little bit more challenging than one would think. And so in this case, when we have these microgels and you interlink them, we also modified them with RGD, which is a very short peptide that binds to the integrants on the cells, and then cells can attach to these microgels. And here you see that the microgels are quite large. Because they are made via microfluidics, you're limited in how small you can make them. So a microfluidic chip is already difficult below 20 micrometer, like 10 to 20 micrometer, because of the pressure you need to push the fluid through. And so these were made with a channel that is like 80 micrometer. So these are about a diameter of 100 micrometer, which is much, much larger than these cells. So the cells will first start to cover the microgels. And once the microgels are covered, then they start filling up the pores. And then we form like more denser tissue. And what we also see is that we don't get a necrotic core. What you normally get if you grow dense tissues in vitro, you have a problem with diffusion of nutrients and oxygen, and you get like a dead apoptotic core in the middle, which we call like this necrotic core. And yeah, so this is something we can avoid by these microgels, because the microgels basically function as a pseudovasculature to allow for diffusion of molecules to the cells. And as these pores are not really larger than 400 micrometer, we don't see any cell deaths into these constructs. But I mentioned that these are large, right? So we also looked into a different technique to make them smaller. And this is an in-mold polymerization technique, which was inspired by the print technique, which was originally developed by Joseph De Simone in North Carolina, but he's not in North Carolina anymore. I should change this. But so he was making particles with this method. We wanted to make soft microgels. And in order to make soft microgels, you have lower polymer concentrations. And to avoid evaporation of a solvent or water, we actually add a non-reactive liquid polymer filler to avoid that. So this way we can make very soft microgels, and then we wash out the filler afterwards because it didn't react with the network. And then you can really harvest very monodisperse microgels, also now all the way down to 0.5 micrometer. But the disadvantage of this method is it's a batch process. So it's very time consuming, and it's not like a continuous production like what you saw with the microfluidics. But if you use these, you can now make very thin microgels, and then also change the aspect ratio, for example, from 5 to 20. These are now 10 micrometer width and different lengths. And you also see that you can change the pore size depending on the aspect ratio of these microgels, and also the overall void space. And this overall void space here is like 90% in the case of the highest aspect ratio, which is very large. And this is because the microgels are very thin. So we're not adding a lot of synthetic material to have a lot of free open space for the cells to grow. And so now these microgels are about the same size of a cell. So you also see that cells now interact very differently, right, with these. So they immediately cover these microgels and bridge the microgels, and then fill up the construct within a week, like the construct is completely filled. But it's still a batch process. So we were then thinking, how can we make these very thin microgels, these rods, in a continuous way, like microfluidics? And so we wanted to find a way to break the limits of microfluidics. And so for this, we decided to make, instead of droplets, to make a very thin jet, which is now 10 times thinner than the channel diameter. And as this polymer solution cross-links with light, we thought we could oscillate the light. So we would have light on, crosslink, light off, no crosslink, on, crosslink. And so the hypothesis was that we would then have parts that are cross-linked, which would form the microgel, and parts that are not cross-linked, which would just dissolve. And so first you see here when the light is on the whole time, you get a hydrogel fiber. When you then oscillate, initially we got these weird trumpet-shaped things that whatever they are, not what we wanted. And this was because we realized the jet wasn't stable. When the light was on, we got like a traffic jam, so to say. And then we added again like a non-reactive polymer filler to increase the viscosity of the jet. And then we got like a system like this. And we realized that these peaks sticking out were actually our microgels. And so when you then look at the product, you see these microgels that are now 10 times thinner than the channel diameter that we used in this case was 80 micrometer. We now are downscaling this to 20 micrometer, and we can now make microgels that are 2.5 to 3 micrometer in diameter, which is a lot more challenging because you have problems with oxygen diffusion, which inhibits free radical polymerization. But that is actually a study which we are just submitted now. So hopefully this will come out soon. Okay, so when you now want to change the length of these rods, you can have the laser, the on time, longer or shorter. And when you have it longer, you have these very long microgels, and when it's short, you have them very short. So you can really play with this. And so you can look at these strands like polymers, but now they're much bigger, right, than a molecule, but they have a certain molecular weight or length, or they have a certain stiffness or persistence length. You can really kind of compare them, but it's just a very different scale. And then you can also interlink them for these 3D constructs. So the constructs I showed you so far were always chemically interlinked, and then also we added the cells on top. But we also realized, together with the group of André Pich, that if you mix microgels with cells, and in this case, these are actually spherical microgels, that if they have this RGD that the cells can attach to, the cells will grab these microgels and then just assemble them themselves into a 3D construct. So they're kind of their own architecture of their own house. And you see in the videos like how the process happens. And depending on the ratio of the cells and the microgels, we consistently get or this ellipsoidal shape or this more triangle shape. And so also with the stiffness, we can like control this in which shape they would form these 3D constructs. So this is something we are still investigating further, also how to control this, but it will of course be interesting if we don't have to chemically interlink the microgels, and then the cells can just do the job and be the glue in between the microgels. Okay, so moving on to the second part, this directional guidance, we want to align them in this case. So we want to have something you can inject, but form an oriented structure. And this was originally motivated by spinal cord injury. And so people with spinal cord injury are paralyzed below the site of injury, and there's really no treatment available. People then realized in the 80s that you can stimulate nerves again to grow, but you also want to guide them in the right direction. And so just to give a little bit of a recap or like an overview of spinal cord. So you have like here the brain, and you're sending signals via the spinal cord to the rest of your body. But if you have an injury here, then because of a car crash or a sport accident, you form this kind of cyst with a lot of scar tissue that builds around it, and a lot of inhibitory molecules that basically form a wall for the nerves to be blocked. They cannot grow anymore. So you need to have solutions to degrade scar tissue, but also to then form a bridge locally to guide regenerating nerves across the injury site. And this is what we are working on. So we have a material which we call anisogel, which then has these microgels that magnetically align to guide regenerating nerves back across the injury site. I will explain this a little bit later. First, I would like to show you how this actually looks. So a patient with spinal cord injury, and you see how the bone crushed into the very soft spinal cord, that you see that the normal healthy spinal cord with the gray and the white matter, now you get this big cavity with like a very inhibitory environment for nerves to grow. And rats are actually a good model to test this, because rats also form this cavity. So mice don't. Even though mice are sometimes used, they don't really form this cavity. And so you see here that if you do an injury, and this is usually with a controlled weight drop, that you get like, first you get a lot of inflammation, and then this cavity starts to form, and also the scar tissue starts to form, then blocking the nerves to regenerate. So an important question we also are studying now, and we do this work in MtGap, which is a project, a very big project in Canada, who are using our materials also and testing them in animals, is finding the right time to inject the gel. Because if you do it too early, maybe there's still too much inflammation. If you do it too late, there's too much scar tissue, and you can't really overcome this anymore. So currently it looks like 4 weeks is a good time to inject the gel, and I just wanted to show you how soft it also is. So this is a very thin needle that is used to actually inject these gels. Here is just like on a surface, but you also see like how soft this is, and this is really needed for then the nerves to be able to grow into this material. And to show you again how the system works, so it's called anisogel, anisotropic hydrogel. We also patented this technology, and it's a 2-component system consisting of magnetic rods, which so these microgel with iron oxide nanoparticles inside, and a polymer solution around it, which then we then add a magnet, so the magnetic rods can align, and at the same time the surrounding polymer solution cross-links into a network to keep the aligned rods in place, and then you can remove the magnet, and then the whole structure is fixed, and then you have these oriented microgels to then guide the nerves across the injury site. And so, of course, we tested this first in vitro, and so you see here how the microgels are orienting, and these are very thin, so these are made via this print technique that I showed you before. And you see that if you optimize the system with the right amount of microgels and the right stiffness and a gel around it, which is good for nerve growth, then the nerves actually follow the alignment and grow in an oriented way. And so this is a chick dorsal root ganglion, which we take from little chick embryos, and then you can take the cluster of nerve cells, and there's also supporting cells. You can put it in the hydrogel and then look how are the nerves extending into this material. And so, one of my students, Susan Babu, she was actually part of the Max Planck School. She did a very systematic study looking at the concentration, the dimensions, the stiffness, if these microgels would be better, if we would also modify them with certain peptides, or if we would actually also cross-link the microgels to the surrounding network. And I won't show you all the data, I will just tell you she optimized the dimensions to be very thin, so like 2.5 micrometer. I will show you later why. And the concentrations, she actually showed that in this stiffness range that we could make them, there's no difference. So the softest one is even much stiffer than the surrounding gel, so it did make a difference. If you modify the microgels with bioactive molecules for the cells to attach to, we had an opposite effect of what we wanted. We had a lot of growth, but the cells would be attracted to the microgels, and we would lose the alignment partly. So they would grow kind of in the wrong direction towards the microgels instead of along the microgels. So that was kind of okay, don't over-engineer the system. So we only have cell adhesive peptides in the surrounding gel and not on the microgels. Everything is made from polyethylene glycol, which is clinically approved to use into the clinic. And if we couple the microgels chemically to the matrix, we have no alignment anymore, so or very, very little. And so this was also not what we expected. We expected an enhancement of the alignment. And this is likely because it's locally changing the mechanical properties too much that the nerves are just not likely to grow as much. We also had less growth of nerves when we did this. So we now know we just should embed them, not link them to the matrix, and the microgels should probably be inert, and then the surrounding gel is the one that provides bioactive peptides for the nerves to grow. So yeah, here you see why they have to be thin. If they're 5 micrometer wide, the growth cone, if they bump into it, then they will be kind of stopped. If they're thin enough and the growth cone bumps into it, they will just kind of slide along the microgel. And we saw this at multiple places. So these are really representative images. And we saw a significant increase in growth rate into the gels when we had the thinner microgel. So this is really the ones we are now testing also in vivo. And I can't show you the in vivo results yet, but we have some preliminary results where we do see nerve growth into these gels, but we need to further optimize it. And so it's a little bit too early to actually show you this. But yeah, we're currently optimizing this. And then once you have a lot of nerve growth into this gel, you also need strategies to get them out of the gel and back into the cord. And for this, you also need to create some growth factor gradients to have them grow back into the healthy part of the spinal cord. So we call this a candy store effect, or like an oasis effect, that they're just too happy then in this gel and they don't want to leave anymore. So that's also a problem. So yeah, the last part, which is a little bit shorter also, I want to show you how we bring movement into the system. And there we use a different polymer. We use thermoresponsive polymers that would collapse at a higher temperature and would swell and take up water at a lower temperature. And so to be able to have this switch around 37 degrees, because we want to do this under physiological conditions, we optimize the ratio of 2 different monomers, NEPAM and NEAM, 60 to 40%, to then have the right transition in media, because salt also affects the transition temperature. So compared to water, there's a big difference. And to quickly switch from the swollen to the collapsed state, we can't just change the temperature the whole time. So we incorporated gold nanorods. This was inspired by work done by Professor Martin Muller here, who was before at the Institute. And to do this quickly, you can then put this gold inside, and they will convert near-infrared light to heat locally. And they do this very quickly. So now you can actually get this actuation very quickly, up to 10 hertz actually, but we see most of the effect on cells when we use 1 hertz, which is a frequency which we have a lot. When we move, when our heart is beating, this is like a very physiological relevant frequency where we also see the biggest effect on the cells. So on this image, you have to look very closely. It's only like this tiny vibration. This is a hydrogel film now, which is patterned with grooves, and then we put cells on top. We have to modify it with fibronectin so that the cells can grow. And it's very little. And the reason why, and this is a disadvantage of the system, is because we can only really change the temperature between 36 and 39. We don't want to grill the cells, right? So we really try to stay in this temperature range. And so there the change is not so big. And so we only have a small amplitude. So we're working on trying to increase this also. But we do see an effect on fibroblasts that are growing less, that are having more focal adhesions, that have mechanosensitive proteins that shuttle to the nucleus. We see myoblasts that are starting to differentiate into muscle cells. We see that mesenchymal stem cells, when you actuate them without any differentiation factors, seem to differentiate into bone cells purely because of the actuation. But I won't show you all these results because of time, but I added here the papers, and actually I should update this because this already has, of course, like a page number by now. This was in 2023. But so you see that this is 2D, and before I showed you a lot of 3D, and so we also wanted to make these microgels of this material so we can incorporate them into our 3D structures. And so here you see 2 videos. One is this beading microgel as an alone standing microgel. You could potentially also interlink them. or you have a microgel that is now fixed to a surrounding gel with cells inside, and then all these cells will be affected by this beading of this microgel. And so this is ongoing work. And so these microgels can also be made via microfluidics. And instead of just using monomers, you can also use prepolymers. And we also noticed that if you would change the temperature during the cross-linking with the UV lamp, because this is a thermoresponsive polymer, the polymers can collapse at higher temperature, so you get phase separation, which you can then fix by cross-linking it. So you do basically the cross-linking and the phase separation at the same time, and then you get very different constructs. So you can get microgels that are more opaque, more opaque or more transparent, rougher versus smoother, very different stiffnesses, whether you use monomers or a 3-arm star or a 4-arm star, and these are made via free radical polymerization, also actually via raft. And and then by changing the temperature also you get very different structures because of this phase separation. And just to show you also some fluorescent images, this one was actually hollow. So this looks like almost like a golf ball, and it's like hollow inside. So very interesting structures by playing with these different properties. And then we also measured the stiffness again via nanoindentation, and you see for the monomers, if you gel at a higher temperature, so with phase separation, they're much softer. For the star molecules, you actually get stiffer structures if you have the phase separation. And interestingly, with the 3 and the 4-arm precursor molecule, you get a much steeper transition. So this means that between 36 and 39, which is the area we want to actuate, you may have a bigger amplitude and a bigger effect on the cells because you started from a different precursor. And then if you have these microgels, you can modify them again with bioactive molecules, and then cells can grow around these microgels, and then you can start, let me go back. The video here. So then they start actuating, and then you could mimic, for example, also breathing or other mechanical forces on epithelia, and then see how this mechanical forces is affecting also the cells. Okay, to just wrap up, I wanted to show you this instrument that we recently acquired, which is this robotic system, which has a pipetting, an automated pipetting system, an incubator, a high throughput confocal microscope, and a robotic arm, which can basically move the plates from one place to the other place. So the idea is to make the construct, feed the construct, stain the construct, analyze them, like all in an automated high throughput way. Of course, we will have to go step by step in order to do this. I won't play the whole video, but here you see now the arm picking up the plate from the incubator, putting it onto the pipetting system, and then you can start pipetting. And here is the confocal microscope, and you can put it into the microscope also automatically. So it's a closed microscope, it's a spinning disc, so it's very fast also. Okay, then just to show you a little bit of the bioprinting we do. So we're in a larger EU consortium to print liver constructs, and there we also made a PEG-based synthetic ink with channels, and then together with spheroids so that the spheroids can grow. And then we found that depending on the cell ratio in these spheroids, they could become hollow, and then the endothelial cells would line the structure. So we think if you can bring these spheroids very close together, that you could start forming these lumens, and you see fluorescent beads in there to show that it's actually a lumen. And this is something we're now further developing to also try to really grow blood vessel structures into these constructs. And then if you want to combine bioprinting with magnetic fields, we call this magnetic field assisted bioprinting, you can now in different layers get different orientations. And this is, for example, interesting for cartilage, which has different layers. The bottom is like oriented like this for load bearing, and then on the top they're like this because of the friction that they want to reduce. So yeah, this way you can also align these rods as you print. And so, or you change the magnet during printing, or we had a very nice collaboration with the group of Jérôme Crassus, where instead of putting spherical iron oxide particles, you can also pre-align ellipsoidal nanoparticles into the microgels during the production of the microgels. And if these are now aligned perpendicular to the long axis of the microgel will align perpendicular to the magnetic field and not along the magnetic field. And so now you could print with one magnet and then change the microgels as you print and get different types of structures. So you can really program the angle of orientation of these microgels. So with this, I would like to summarize. We make a lot of these colloidal and polymer precursors that can be injected or pipetted or printed, and that they can in situ cross-link to form these hydrogels. We like these rod-shaped microgels. They can be linked together and really create bigger pores for better self-infiltration, but also cell-cell interaction, which I think is very important to very quickly have cells interacting with each other. Then we can also add these magnetoresponsive microgels, so we can orient them and also direct cells in a certain direction. And then we would like to also actuate them in order to mechanically stimulate cells and see how this affects also stem cell differentiation or other biological processes. Yeah, and so with this, I would like to, of course, thank my group. They're really the ones doing all the work in the lab. And so I'm very fortunate to have very creative students in our group. And then, of course, collaborations, collaborators. We work with a lot of different groups together, which I think is very important to combine expertise to be able to achieve more. And that's also, I think, the vision of the Max Planck School, bringing people together to form these networks. And of course, funding, because research is very expensive and we need a lot of funding for this. And of course, also you for your kind attention. And I would be very happy to take many questions.
Thank you very much for this exciting talk. And first questions are already coming in. But for everyone, I mean, please use the question and answer bubble on the bottom of Zoom. Type in your question. And I think the very first one, this just came when we were having our pre-discussion actually to the presentation. So the student was asking how to prepare better for a research career. You want to say something about this before we start then with the more topic-specific questions?
Yeah, I think it's such a hard question, right? But I think what is very important, and I'm not saying this is easy, is to really form your unique, Alleinstellungsmerkmal of Deutsch, how do you call it? Uniqueness profile, basically, what you are known for, and that you find something, and this is of course the hard part, where you see, okay, this is really a gap that I can fill, maybe by bringing in certain expertises, by combining certain expertises together, something you think about that maybe other people haven't thought about before, and then really work on that you're known for that specifically. And of course, it is very important to publish well, right? And I think it's also as a PhD student, sometimes hard because things don't always work well, but just keep going. And the more things you try and the harder you work, and I'm still from that generation that believes that hard work actually pays off, that it's not only luck and only being efficient and productive. I think also if you work hard, there's just, you increase the statistical potential to actually find something. And that happened to me in my PhD as well. I had these aha moments after months or like a year where things weren't really working, and then suddenly it worked, and it's like this euphoria moment. And I really wish that upon everyone. But yeah, so try to find this niche that you can really make your profile.
Thank you very much.
And maybe one way to do this is to pick a postdoc which is different than your PhD. And then see how you can link different fields together.
Broaden out. Yeah.
Thank
you. Okay. Then the first question related now to the talk. Can the microgel we aim to use in the spinal cord also be used in the brain tissue in the future? can people living with brain damage by tumors or similar causes benefit from neuroplasticity more efficiently with the help of microgels?
Yeah, I think so. And actually, Tatiana Segura, who uses the spherical microgels, is doing that. So she's actually, her biggest model is actually the brain. And they also do skin. And actually, her microgels, she calls it MAP scaffolds. There's a spin-off company now, and it's now for skin regeneration already in, so this is now going into humans. So she actually was able to do the whole translation, which I think is very admirable. But I know she's also interested in rods, and so yeah, hopefully we can maybe work together at one point, and she could then compare the rods with the spheres and look how cells grow differently if they have more space.
Mm-hmm. Thank you. The next question is, you said you can orient the microgels magnetically.
Mm-hmm.
How can you prevent them from deviating? Do you need to fixate them somehow?
Yeah, so that's what the isogel principle is, that you have a two components. So one, you align the microgel, and at the same time, you have a gel that cross-links around it. And so you basically fix the orientation by the surrounding gel, because the surrounding gel has much smaller pores, these nano mesh pores, compared to the bigger rods. So they won't, you can even flip the magnetic field, nothing will move. But you just take it away and it's fixed.
Thank you. And the next question for the gold nanorods utilized in the actuating hydrogels. They will be inserted in the damaged tissue, but after the gels have degraded in the bodily fluid, how would it be ensured that the added nanorods are processed out of the body? Is it naturally done by the body, or would they stay in the localized region where they were inserted?
Yeah, so maybe to correct the actuating microgels we mainly use for in vitro systems. For example, we want to make mini heart tissue, and then we want to see if the actuation would better differentiate these induced pluripotent stem cells into more mature cardiomyocytes because of this actuation that we can bring in. So that's something we're currently studying. We're not really immediately doing this for in vivo, although one could think, okay, maybe actuating may help the regeneration process as well. But similar to the magnetic nanoparticles, so once the microgels would start to degrade, and then we have to make them degradable enough because currently they're not yet, then the macrophages would start to digest everything. And we just have to be sure that we stay below toxicity levels, which we do. Actually, to align the rod, you don't need a lot of iron oxide nanoparticles. So that's a good thing. But we have to, in vivo, we will now look at the long-term effects of this material.
Okay, just coming in the next one. I have a question regarding how you managed to assess the mechanical properties of your microgels. You mentioned nano indentation.
Mm-hmm.
Sorry. But were they embedded in some other matrix?
Yeah.
Also, why did you choose gold nanorods instead of other anisotropic shapes like stars? Finally, regarding the mechanical hydrogel studies, did you study migration?
Okay, that's a lot of questions.
Okay, maybe I'll can read the first one again, and then we go.
No, I remember the
Okay,
first
good.
was the mechanic. If I'm wrong, you just correct me. The first
No.
one is the mechanical characterization. So
Yeah.
If they're large enough, we can do nano indentation. If they're too small, like the print ones, we have to do colloidal AFM. And so nano indentation, we bought this instrument Pavone from Optics 11, some advertisement maybe, like, and but we don't embed them in a gel because then we don't measure the microgel. So we actually do have something where we stabilize them on a surface by interacting with a surface, but then we measure on top. So just a compression via this nano indentation. You have different sizes of colloids to do this as well. So when smaller, colloidal AFM, yeah, and when larger gels, we use dynamic mechanical analysis of rheology. That's kind of the range. Then people could do also these optical tweezers. We just got this instrument at DBE, but I don't have any experience with this. But I do know people also use these optical tweezers. Second question, why gold nanorods? We wanted something that would absorb light at near infrared light. And depending on the shape of the gold nanorods, you can tune at which wavelength they absorb the light. And so we wanted near infrared because it's harmless to the cells. We didn't want to use UV or something like this. And it can penetrate also deeper into tissues. So that's why we chose that one. And we modify it with chemical groups that the gold nanorods are actually interlinked to the network so that they don't leak out of the system, that they stay inside. Yeah, so we synthesize them ourselves. And then the last question was about migration of cells. There I didn't really understand how do we measure that, or how do we...
So the question was regarding the mechanical hydrogel studies. Did you study migration?
But the mechanical hydrogel, which one? They all have a certain mechanical, the actuating ones, or anyways,
maybe Maria could specify
we
this.
do videos, right? We do live imaging and we make videos and we look at migration. Yeah, okay,
The
because
actuating ones.
I mentioned the first paper we published with the actuating gel was in Nature Communication and in 2019, I think. And there we see indeed that fibroblasts, I always compare it with a baby on a jumping castle, and then the big kids are jumping and the baby's afraid and they're like holding on to the surface. And they really do, like they migrate less, they produce more extracellular matrix to attach, they have more focal adhesions, they have this MRTFA, which is a protein that shuttles from the cytoplasm into the nucleus only when you actuate. And MRTFA holds G-actin. And when it goes to the nucleus, it lets go of G-actin and it forms F-actin. So it's basically creating more cytoskeleton for the cells to be stable and really adhere to the surface. And so because of this actuation, the cells seem to be like holding on to the surface. But we did a lot of video time lapse to look at migration.
Thank you very much. Then the next question is for the droplets cross-linked to obtain rod shape, and the cells are added later on the surface using RGD motifs. Is it possible to rather embed the cells inside rods in the beginning, which would allow the cells to have their own microenvironment and they would assemble as the cells interact?
Yeah, it's possible. We actually did that and also published this in Small. So you can look into, I think it was all 2017 or something. And there you need a chemistry which is degradable. So there we use a Michael type addition, so two components that come together, and then we had an MMP sensitive crosslinker that the cells, when they would produce MMP, they would degrade the crosslinks and they would have space. And then you see that the cells are really, and they're very soft also, otherwise cells don't grow. And then you could see that the cells were aligning into these rods microgels, and there were a few cells per microgel, right? It was not just one. Yeah, we have, and then we have another paper also where we put cardiomyocytes together with a group in Japan where we coated cells and then put them in microgels, and you see that then the cells are beating also. And so you could think about using these microgels as a delivery system for cell therapies in the body, because if you just inject cells in buffer, a lot of them will migrate away or they will die. But if you would inject cells in these microgels, you would give them already a supportive environment and you would avoid that they would just migrate away. So I do think that's also something that could be promising in the future, but we're not really working on that so much.
Now there's another attendee posted a couple of questions, but I think to sum it up a bit, it's about where you see the future for biomaterials in the future. And so where are trends going, and how biomaterials will evolve, or what maybe they have the most impacts on in the future, and where you see also the advantages of machine learning tools for this kind of research and in tissue engineering.
Yeah, I think for the design, I think machine learning indeed can play a big role. They're also, especially also for the analysis of how cells interact with cells, and then via all these images with these high throughput systems, then I think machine learning will also come into play. To then do kind of reverse engineering and design these systems. But yeah, we have like a new big proposal here in Aachen, which is like our excellence cluster, transformative medical materials. And so we are really looking into how we can design materials that really communicate with the environment. So if something happens in the body, that the material can adapt to this signal. And this can be done for many different applications where we really want to have materials that have programmed information in them, like when something happens that they can respond to this by delivering a certain factor or by degrading in a certain way, which would then kind of make the implant bigger, like one of my colleagues, for example, would like to look at growing implants over time with kids. That would be like one of the challenges also. So materials that are smarter than the ones we have now, because currently I think we're kind of at this plateau also of the materials that actually go into the clinic and what they can actually do. And I think it's because they lack this really interactive communication with the body. So responding to internal triggers, or if we see via imaging that something is needed, that you use an external trigger like ultrasound or magnetic field to change the material or to release something that is needed at that time.
Thank you. Now I think we come to a little more technical question. In your actuating hydrogels, you mentioned NiPAM and NEAM. How did you overcome problems with cells detaching, since NiPAM has
NiPAM and
properties?
NEAM. Yeah, we use NiPAM and NEAM, and indeed you need to, we use a sulfosamfa in order to then couple fibronectin to the surface, or we add again the same monomer that I showed in the beginning, this GMA, you can cross-link this into the network, and then you have an epoxy on the outside, and then you can couple anything with an amine, and all proteins have amines, you can have peptides with amines, and then you can easily modify the surface with bioactive molecules for the cells to attach.
Thank you. And then the question about the modules that you are using in all the different variety in your group. Yeah, are they all, or how many of them are you think are promising for the usage of bioprinting or in bioprinting?
Yeah, I guess yeah, that's a good question, but I think a lot of them are actually. But when people talk about what is needed to have a bioink, a lot of people say it needs to be extrudable. But I would already disagree, because the material we actually printed is like water. It's like a liquid, but then you can use like a sacrificial mold to keep the liquid inside with channels. Like that's what we did. We had a mold and we had channels that we printed that we could then dissolve, and then you fill it with the liquid PEG solution, and then you cross-link it. You let it cross-link, and then layer by layer you form a very stable construct like you see here. And then because sometimes you need to print a little bit stiffer to have a stable construct, so we countered that in this publication by adding a faster degrading crosslink. So we print a bit stiffer, but then it degrades quicker for the cells to grow. And this is how we kind of overcame this challenge between like 3D cell growth and printability. But I don't think it needs to necessarily be extruded. You can use a light mask. You can use all kinds of techniques to get structure into these inks.
Thank you very much. So at the moment, I don't see any further open questions. And also, I mean, we're at the end of the time, so I think that fits quite well.
Yeah.
So thank you very much for the presentation. Thank you very much for the great discussion. Yeah, thank you everybody for being in the audience and listening and discussing with us today. We're looking forward to see many of you again during our next MtL Lecture Series, which will be on March 12th, together with Christine Selhuber-Unkel here from the University of Heidelberg.
That's going to be a great talk as well, so I would highly recommend that.
Yeah, I guess absolutely.
I also want to thank everyone for joining. I mean, I don't see you, but thanks for being there, and it was a lot of good questions, so thank you. And thank you, Jan, for the invitation.
Yeah, very, very welcome. Yeah, always happy to have you here and present your current research. Thank you very much. Have a great afternoon, and see you soon.
Bye. Bye.